Okay, hello, hi. I'm Jason Williams, and I'm coming to you from Cold Spring Harbor Laboratory in New York. Welcome. This is our quarterly webinar, CyVerse Get Started, and we hold this webinar live every quarter so that we give an opportunity for people who have just signed up for their CyVerse account to get to know a little bit more about the platform. And one of the reasons why we've always liked to do this live is so that we can take your questions. We're using here the Zoom webinar format, which gives you the ability to go ahead and ask those questions, pardon me, and I will go ahead and answer as many of them as I can during the presentation. I have a few slides that I prepared for you to give you some background, and then I'm going to go through 3 demos that talk about how we do data sharing in CyVerse, how you can use the Discover environment to do analyses, and also how you can use the CyVerse Cloud. So all of these resources are free. Hopefully you've had the account maybe even for a couple of weeks now and have had a chance. Many of us are also thinking about how we can be more effective at using research, computing, and software resources from home for research or for our education or teaching. So any and all questions that you have, you can go ahead at any time and use the Q&A. That's the recommended way to do this. Use the Q&A button and send me the question, and I'll attend to as many of those questions as we can. This webinar will also be recorded, so you'll have a chance to review the material after the webinar is complete. So with that said, I see many of you out there in the audience. I'm going to go ahead and start sharing my screen. And I have a couple of slides to get us started, and then once we go through those, I will go ahead and also give you a chance to see some live demos that I'll do. Okay, the first thing I want to remind everybody is that everything I'm covering and more is available to you in our documentation. And this is available at learning.cyverse.org. So if there's anything that you miss or anything you really want to go back to and look at it in detail, perhaps more detail than I'm going to cover it, make sure to go ahead to learning.cyverse.org, and then you'll see a whole bunch of information as well as upcoming events. Obviously, our physical in-person events are not happening at the moment, but we do have the ability to do virtual events, and we have a lot of support mechanisms. In the end, if you need something and you just don't see it, let us know, and we'll try our best to help you. So let me start with just a few slides. It's going to take all of 5 minutes or so, just to give you a bit of background. Our vision as a project is really to help transform science through data-driven discovery. And we have more than 70,000 at this point, probably close to 80,000 users, which has contributed to the petabytes of data that run across our system and hundreds of publications, courses, and discoveries that have been enabled by the CyVerse resources. CyVerse as a project has been around since 2006, where that was when the first proposal was put in. It was actually funded in 2008, actually to serve the needs of the plant science community. From there, the project was successful with its launch of tools. It was renewed again by NSF, and we kept evolving and even changed our name to CyVerse to represent that we're really serving a much larger audience than just the plant sciences. And we had our last funding renewal by NSF in 2018. So we certainly want to give credit because we are an NSF-funded project, and that's a key take-home message. We're not a company. We are an NSF research project, although we're really tasked with science enablement. And we're here to support you. So we're your colleagues, we're your collaborators, and it's more than $115 million in NSF investment. So it's really things that you don't directly have to pay for in order to take advantage of. It's freely available to the community. It's really open to spur national and international collaboration. And one thing we do hope you remember is to acknowledge and cite us in your publications. That's one of the ways that we demonstrate impact. So CyVerse is primarily a cyber infrastructure project. What does that mean? Well, in some sense, it's things that you're already familiar with: data storage, software, high performance computing, and even people resources who have that expertise to help you when you need help. It's really taking those things and organizing them to systems that really have been designed to allow you to tackle research and educational objectives that you might not have been able to do without that kind of resource. We're really community focused, and so everything that we built has been built around the needs of the community. So when you come to CyVerse, and as you'll see a little bit in our demos today, it's about platforms, tools, and data sets, storage and compute, as well as training and support. And today is another example of that. Another take-home message is that CyVerse is built for data. So no matter what your system, whether microbes, plants, animals, medical data, ecological data, or even the different data types, whether sequence data, which is still the largest driver of life science data, but also images, very much so, and other data types you might have, CyVerse is really built to accommodate all of those. This is a very broad overview of some of our platforms and systems. Most of us who come from the biology perspective might want to work up at the layer of products, which are the forward-facing interfaces, usually web interfaces, to the different systems that we provide. But if you're a bioinformatician or you work with bioinformaticians or a research software engineer, there are whole layers of systems which you can customize and extend. So everything from our search APIs to APIs that allow you to work with the actual HPC computing resources to consume them in a way that's very custom. So you can decide which way or what angle you want to look at it. If you are a bench biologist, you can get your research done. But if you're also a software engineer, you can also customize and use CyVerse in very creative ways. Clearly, genomics has driven the needs of big data biology. So when you come to CyVerse, you're going to find tools for a lot of the things you'd expect. To be able to go ahead and do assemblies, RNA-seq, variant calling. Those are things that we currently support, of course. But there's more than that because bioinformatics is more than genomics. The other thing we want to recognize is that we are more than just one organization. In the US, the original CyVerse is a consortium between University of Arizona, Texas Advanced Computing Center, and Cold Harbor. We're also a virtual organization, so there's CyVerse UK, and no, there's not a slide for it yet. There's also CyVerse Austria and other installations that are supported in various different ways. So what I want to turn my attention to now that I give you a little bit of a history lesson about our project, very proud of it, so we did want to share that, is really what the actual components and products are so that you have an idea. And like I said, please go ahead, this is live. If you're watching it right now, unless you're watching the recording, which will be posted to YouTube, go ahead and use that Q&A button as you have questions. What I want to take you through now are the individual products, some of the most important products, several of which will feature on today's webinar. We have had this vision of our system really as sort of the Lego blocks of bioinformatics and data science biology. So we've built many of the pieces that you need. And a lot of times we have a set that's already put together, but oftentimes you have something custom. And so we've built 85, 95% of what you need, and you may want to customize that sort of last mile. Okay. So here is one of the major components. First of all, the data store. So you need to be able to store your data on CyVerse, and every user has an initial 100 gigabyte allocation. You can have much more than that if you're operating on the level of you need terabytes of data. We can service those requests. We do ask that you fill out some forms that you'll find on the CyVerse website so that we can track that it's being used appropriately. Those data are automatically backed up at various locations. They're secure, and we've made it easy to upload, download, and share. And I'm going to demonstrate some of that in the first demo that I do in just a moment. The discovery environment I'll also cover in a demo, and this is an easy to use web interface where you can run almost any bioinformatics application that perhaps ordinarily would be run at the command line, but you can actually run it without sophisticated knowledge of Linux per se. It's integrated with our storage and our high performance computing, and it's also extensible, so you can add your own applications as well. One of the cool things about that, which I'll also show, is this thing called Bice. It's a subcomponent of the discovery environment. So if you're used to using some of the popular data science tools like Jupiter or R or Shiny, you can automatically launch and configure your applications through that system. We're also going to show you in our demos today Atmosphere, which is our cloud computing resource. It's one of the earlier cloud computes. It's fully customizable for life sciences and is actually directly available to you. You can work with hundreds of virtual machine images, and we'll explain that once we get a little bit into that area of the demo. Now, there's some things that I can't show you very easily, or actually, in this case, for sake of time. We do have a full set of science APIs that allow you to customize the way that you consume. So if you're a developer and you want to do something that's just not on the menu as one of our products, that's also possible through the APIs that we make available. DNA Subway is a really fantastic resource for the classroom. So if you're teaching and you want to talk about genome annotation, we have a wonderful set of experiments, wet lab and dry lab, on DNA barcoding, also on RNA-seq, and also on metabarcoding and microbiome analysis. Those are all available in simplified but robust classroom-friendly workflows, where you can work with real data sets to work on real research problems, but in a way that's really robust for the classroom. We won't show that in the demo, but we have lots of things online where you can learn more. And if you check out our YouTube channel, you'll also find some webinars that we've done over the last several months. BISC is for working with image data. If you have any one of hundreds of image types from microscopic to microscopic, there are customized ways to organize and even analyze those type of data. And that was a very whirlwind, very quick tour, but now I'm going to give you a little bit more in detail for some of these. And the first demo that I'm going to do is going to talk about how to get your data into CyVerse. And like I said, this is live, so at any time, go ahead and hit that Q&A button and send me a question, and I'll happily respond to everything I can. So the first demo I'm going to talk about is the data store. And with the data store, you can store any type of file related to your research. And you can also move files between different CyVerse platforms. For example, I mentioned that we might be using the cloud atmosphere, or we might be using the discovery environment, or we might be using something else. So all of those platforms are largely connected to the data store. I'm also going to talk about how you can automate things between your server, your local computer, and the data store, and how that makes it easy, therefore, to share data with lab members, communities, collaborators, etc. There are a number of ways, these are the three main ones, to access the data store because there are just a number of use cases. So the two point-and-click methods, which I'm going to show, is CyberDuck and the discovery environment. And then there's also one called ICommands, which is a command line version, and they all have their different uses, and I'll go through them briefly, enough to get you started. But again, remember, if you go to learning.cyverse.org, the full documentation can be found there. So the discovery environment, which is actually the last one we'll give a demo on, is great for working with small files in terms of uploading, downloading. It's mainly a data analysis platform, but you do have some file transfer capabilities there. We're mostly going to talk about that because it makes it really easy to share the data through the discovery environment. CyberDuck is really probably the simplest and most robust way to transfer data. That's the first demo I'm going to give. You can drag and drop folders just like you would on, say, Dropbox. But whereas Dropbox is really not meant for very large files, and there's some issues, particularly with versioning and tagging and other things with Dropbox, this is really designed for science and for data sets that we as researchers might want to use. And it's really easy to work on your desktop. So I'm going to show that one. And then ICommands really gives you full flexibility. You can do all of this at the command line, which also means that you can write scripts. You can synchronize data sets. There's a whole bunch of stuff, but you do need a little bit of Linux in order to do that. So I'm going to go ahead and give you a quick demo of those platforms. In order to do that, let me just get things here. 1 second. I'm going to switch what I am sharing. And I'm just going to go ahead and 1 second. Haven't done this on this computer. I'm going to go ahead and share my desktop. And on my desktop, besides these slides, you can actually see this application, which is CyberDuck. So I'm going to go ahead and open up a new browser just so that you see what it looks like. And this is the application. Unfortunately, I can't make it necessarily bigger on your screen. You can view this full size. But also, let me point your attention to where this comes from. If I go to the CyVerse homepage and click on Learning, and let me go to learning.cyverse.org. If you go in here and search for or browse through our platform guides, I search for the data store, or even if I searched, which I should have, for CyberDuck, let's go this way. Wrong one. Datastore guide. In fact, let me share that directly so you can see it a little bit more easy. What you want to do is you want to go, when you're searching for the Datastore guide or going to it, you want to click on CyberDuck. So this is going to take you through the installation process of working with CyberDuck. And the thing is that there is a one-time download that you need to do of CyberDuck. It's a third-party software, which we do not make, but it is free for you to download. And you'll follow these directions, and in all of 5 minutes, you'll be able to configure CyberDuck. So if I go back now to my desktop, when you install it, it'll look like this unit here, and you'll double click in order to access it. But already on my computer, I have connected. It takes about 20 seconds to connect to the data store. And so all of these folders and files here are things that are in my CyVerse data store. And if I want to get one of them, here is a dummy file called very large fasta.txt. I can literally drag it onto my desktop, and a new window will open telling me, well, it goes ahead and it authenticates, and then it will go ahead and do the download. And in fact, here is a history of uploads and downloads. And as you guess, if I have a file or folder, maybe I have a folder that has a couple of dozen FASTQ files, I can go ahead and drag that from my desktop into the CyVerse data store, and it will go ahead and share. So when you're doing transfers this way, you would expect to be able to transfer about 100 gigabytes of data within about 30 minutes on a really good connection. So it's really that simple that you can drag and drop once you have done the configuration, and that information is available to you on learning.cyverse. Okay, so let me show you one other way to do data sharing. I'm going to go back to sharing my screen. Now, this time, what I'm going to do is minimize that, and I'm going to show you the command line version. This is something called ICommands. My FASTA file, I'll just put away. I can actually put in the trash. I don't need it. Let's start a shell that is a little bit more contrast. Let's see if I can do that. That's nice. Okay, a little bit easier to see there. And I'll make it bigger. So let's do this the right way. If we went to back here and we looked back in the same data store guide, there is a guide there on working with ICommands. So ICommands is available for Linux or for Mac OS. If you're using Windows, there is a version that works with Linux. And so for Windows 10, you can install the Windows subsystem. And there's some instructions there on how to do that. But follow the instructions. It does take not that long in order to go ahead and configure your information for CyVerse. And so you just need to follow the directions that are here. And within a few minutes, you will be you'll be connected. Now, I have not, I've already done this. I'm not going to go through the installation, but let me show you a little bit of the commands that you could use. If you're familiar with the command line, then these will make sense. And if I want to list the contents of my directory, it's not ls as you'd expect at the Linux command line, but ils, and hence ICommands. And then all of the things that are on my screen are actual files and folders that we just saw a moment ago in CyberDuck, but that are on my CyVerse data store. And then if you read the documentation, I'm just going to do one transfer. You'll realize what the commands are. So an iget command allows me to download. If I want to download a folder, I'm going to use the recursive option, and I want to see the progress and the time. So I'm going to do that on the VCS folder that I see up here. And I'm also going to put that onto the desktop of my local computer. I'm going to hit enter, and if I haven't done any typos, I see already on my computer there is a folder that's appeared, and it's giving me a readout of everything that it's transferring. And I think there's only 4 files there. So now on my folder, on my desktop, I have that folder that I was able to download from ICommands. And there's a whole bunch of commands that allow you to upload and download. There's commands that allow you to do IR sync, so you can synchronize data between a local computer or local server and CyVerse so that you make sure that you have your data where you need it and in the versions that you need it. There are a whole bunch of other features with ICommands that I can't go into now, but that's certainly enough to get started and point you in the right direction. That with just a few keystrokes, even if you don't know the command line, you can start to use ICommands. Okay, so let me show you one last thing about data transfer or data sharing, and that's going to really move into how do we analyze data with CyVerse. So this time I am going to share my web browser. Okay, and from the CyVerse.org homepage, there's news and stuff going on, but I'm going to go ahead to products and say discovery environment. And from the discovery environment, I'm going to go ahead and click on launch. Okay, so this is the CyVerse discovery environment. I'm going to talk more about it as an analysis tool in just a moment, but for right now, I'm just going to focus on data and data sharing. So if I click on this data window, what I'm going to be presented, the discovery environment is kind of like a virtual desktop, if you will use that convenient analogy. And if I click on the top folder, it's usually going to be my CyVerse username. What I'm going to see is a listing of the files and folders that I just saw, both in the CyberDuck as well as ICommands. And depending on what there is there, I will be able also to click on, some files and preview them and see, what they are. This is a FASTA file. So I'm assuming clicking on it will be pretty easy to give me some kind of text. There may be, I don't know if I have any CSV files here, but you get the idea that you can look at least certain things. I could search, but I'm not going to do very much more. There are the HTML files, et cetera, et cetera, et cetera. And then if you click on these three dots to expand, there are more information. In fact, actually, if I move my window and make it a little bit bigger, you can see when you have modified files, what the type of file is, the MD5 checksum, et cetera. What I really want you to think of for data is not for upload and download in a discovery environment. You can actually do that to a limited extent, but because this is an HTML site, it's quite slow and it's not as robust. But where things excel is sharing. So to go back to the learning.cyverse.org, there is a whole page on data sharing. And data sharing will go through what I'm about to show you now, which is how to create some links and also how to share large files. So if I have an individual file that I want to share, here is a CSV file that I was looking for a moment ago. I'm looking and I have no idea what's in here, if anything. It says it's 2 pages, so I'm assuming, yeah, that I've got some kind of some information here. It looks like some RNA-seq experiment. If I have that file selected and I go to share and I say create a public link and go here and click create, what it will do is create a URL. And if I show that link and copy it, I could take that and put it on a website or give it to a collaborator in email. And when they put that link into their web browser, they don't have to have a CyVerse account. They'll just go ahead and get a page where they can download a file. This is really convenient. It works. There are some limitations. It only works for individual files, not folders. And we don't recommend it for very large things. So if you're starting to get above 1 or 2 gigabytes, transferring that data through regular HTTP can be slow. You can have a failed transfer and have to start over. So we have a little bit better ways to do that. Also, if I have links, because theoretically a link is discoverable on the internet, I wouldn't want to share any data that I consider sensitive or privacy restricted that way. And in fact, one of the best practices is if I'm done sharing something, I can come back here and open up that same data window, but then just go ahead and click on the minus button and it will go ahead and destroy that link and disable it. Sounds a little bit better than destroy. But now if somebody goes and uses that link, they'll get a notification that doesn't exist. So that's a quick and simple way to share at least some data. But if you want to go ahead and share a large amount of data, or if you want to share entire folders, then there are different ways to do that. So here is a folder. It probably has quite a bit of data in there. It really is a min-in upload. And if I go ahead and click share, this time I'm going to say share with collaborators. So share with collaborators does require the person to have a CyVerse account. But the good thing about it is that once you've uploaded something, if you want to share a folder and it contains a terabyte of data, then that person you share it with, they will not have to worry about uploading and downloading, because we're not transferring files, we're just manipulating permissions. And if I want to share that, I could go ahead and enter the name of somebody that I want to share it with. And as I start entering it, I'll get their full name and their institution, so I have a better idea that it really is them. And then I can decide what level of permissions I'd like to share it with, whether it's read, write, or own. Now those permissions, if I go back to the documentation, are listed on our website, and so you have an idea of what they entitle a user to do. Own is the most serious permission because they can actually delete the file, so you want to be careful not to share it with somebody you don't want to have that level. And then the other thing is, if you go to collaborators, you actually have the ability to sort of bookmark people that you commonly shared with, or you can create a team. So for example, if you have a classroom and you have 20 people enrolled in that class and you always want to be able to share data with them quickly, you can go ahead and create a team, and then you can share data with them that way. So it's a really fast way to decide that you want to have a folder and everybody has access to that. And that'd be good for your class, for your laboratory, etc. So those are the main ways to share data. There are some other data features that I do not have time to go into right now, but one of the most fantastic ones, and you can go ahead and learn about it, is the ability to take a folder and apply metadata to it. If you think about it, a folder name can only have so many underscores, can only be so long, or a file name. But with metadata, you actually have the ability to attach a whole other data set. If I were to go just really quickly and show you an example of the level of descriptiveness that you can add, let's see how good I am at remembering where this is. Let's go to CyVerse training. And also, I think it's in quick starts, maybe, it should be in platform guides actually. Let me go back. Datastore. Oh, no, it is here. SRA submission. So for submitting data to the sequence read archive, which you can do directly through CyVerse, it requires metadata be assigned. And so if I click on a folder that I know, if I recall correctly, has metadata, if I click on Edit View, what I could see is that there's a whole bunch of information. In this case, these are descriptive metadata that follow the exact format for the NCBI sequence read archive. I have all of these metadata things attached to it. And the other cool thing, besides being able to apply whatever your choice of metadata standards are, is if I want to go ahead and search, if I click on the Smart Search, I can go ahead and progressively click on the smart icon, progressively add things like metadata. Oops, that's window went behind me there. Let's try that one more time. Okay, metadata is attribute, genus, thou, us. You can come up with any metadata attribute for any of your files. And in fact, if you have 10,000 files, you basically can upload a spreadsheet that contains your metadata and apply those to your individual files. I'm not going to save this query, but just that quickly, here's a little dummy file. But because that metadata, this one is in a place that I don't own, so I'm not seeing it there because I searched just in that place, there are metadata attached to it. So it's a cool feature, but we're going to have to move on for sake of time. But I really would encourage that practice of making use of the metadata. So let me go back to my slides and continue on with the demo of how do we analyze some data. Okay, go back here and find my slides. Let's do it this way. Okay, so we've done that demo. And we'll talk now about the discovery environment. So the discovery environment is a platform that we've just been in for the last few minutes. And what I'm going to now focus on is the bioinformatic applications that you can use. They're integrated with the data, which means that you can see all of the data in your data store and also compute. And although I won't show you how to integrate your own applications today, do be aware that you can integrate them. In fact, they are all Dockerized. So if you're a developer, essentially you can put things in a Docker container and then run it on CyVerse as an interactive app or as a regular application. We'll see more of that in a bit. We talked about data, so I won't focus there. But now we're going to talk about apps and also analyses. So what we're going to do in this demo is we are going to just do something really quick. We're going to take some unaligned DNA sequences in FASTA format and create a multiple alignment using muscle application. So just something that won't take a lot of time, but give you an idea of how this could work. Okay, so I'm going to go ahead to apps. And when I first load this applications, I can see applications that perhaps I have customized or designed. I won't go into it too deeply, but I also can create communities or classrooms, in which case I curate applications that I would like to have quick access to, or that I want my users, my students, my community, I want them to be able to find my community and then know that these are the applications that I vetted. But I can also go ahead and look for apps by topic or by operation. So there are some sorting. There are so many apps out there, it can be confusing, but we give you the communities option to really help you organize and curate. I'm going to go ahead and search for an app called Muscle, which you may not know is one that does multiple sequence alignments. And there are a couple of versions. I happen to know that I want to use the Muscle 3.8.31 by Sheldon McKay. This happens to have a rating, not all of the apps do. If I click on the little three dots, I can get some app info. I can make it a favorite. I can make a quick launch or add some comments. So we give a lot of features for users to go ahead and make their own annotations and decisions and ratings and etc. The app info is useful because it may link to some additional documentation, give you an idea how many people have used it, give you an idea of versions, etc. Any of that information may not be apparent on first glance. But to actually use an application, I click on the name of it. And all applications in the discovery environment essentially look like this. So if you can use this app, you can use just about all of the apps in the discovery environment. By default, my analysis must have a name. It's going to get a timestamp to it, but I'm not going to change that name. I will add comments. I'll say this is a demo. And with any type of point and click interface, the danger is always that you just point and click and you don't remember how you got somewhere. So we really encourage you to leave comments and notes to yourself, and there are multiple places you can do that. This is one of them, so that as you're doing something, you're trying something, you have some record. Sometimes we're a little bit more sloppy with keeping track of our bioinformatics than we would be in doing experiments at the bench. If you're using command line, you kind of automatically have the scripts built in or an output of your history, but with point and click, you've got to fight to do it. So here's how we do it. I almost never have to change the output folder. That's where my results are going to be. But you actually do have the option. So let's say you've created a folder that you share with your lab. You could change this from the default, which is always your analysis folder. And in your options here on the upper right-hand side where you have preferences, you could make your default, you know, if it was me, I could say the Williams Lab folder, and then I would always want my stuff to go there. But I don't have to change it. Here are required, so I need to select input data. So I'm going to browse for a FASTA file. And maybe if this is the last thing I used, it might be, yep, it went straight to this sample FASTA file, which I think is going to be good. Actually, let me use this one in Muscle 3.8.31, because that was a FASTQ file. I'm going to choose the input and DE sample plants. And if you follow the learning.cyverse.org information on the discovery environment, you'll see everything I'm doing is written for you right there. My sequence type I happen to know is DNA. And I can choose resources. Here you get a little bit of menu. I don't have to touch this. The app provider or creator may have already chosen this for you. But in some cases and in some applications, you may know that you need a certain amount of computational power, and then you're welcome to make those selections. There may be some restrictions, but if you do find a restriction is too restrictive, let us know and we'll work with you to change. It's a good time to mention the bottom right-hand side is this sort of live help and live chat. So you can always leave us a message. Even if somebody doesn't answer you within a few seconds, they will get back to you because it creates a help support ticket. Okay, I'm satisfied with what I did. I'm going to go ahead and click Launch Analysis. And I'm going to get a notification at the top and the side that this analysis has been launched and submitted. And in order to see the progress of it, I can go ahead and click on Analyses. And in Analyses, I see a history of all of the analyses that I have done. If I click on those three dots, I can also here do a whole bunch of things. For example, I could update the comments and say, you know, this worked, this didn't work, or this is trying the 2-hour time point. You could just go ahead and leave notes to yourself, whatever you want to do in order to annotate, and that's useful to you. In my case also, if I click on Analyses and click on View Parameters, I get a listing of all of the parameters that I use in this particular app. And I can even save that to a file, like a text file. So I don't have to guess at what parameters I might have used in the past. I always have a record there. And in fact, if I click on this link, I'm not going to do it now, it'll take me directly to the input and eventually when it's done to the output file. So I have lots of ways to sort of trace my history. I see the analysis completed. I'm going to refresh that. I'm going to come back to that in a moment. I also see a question, which I'll get to in a second. But also, I have the ability to relaunch an analysis. So let's say I use an application that had a dozen parameters. Rather than go ahead and launch it from scratch and change all of those dozen back to the way I wanted them, clicking relaunch, and I did that kind of quick. So I check an analysis I've done in the past and click on relaunch. It'll show me that analysis with all the parameters I used last time. And then if I wanted to change this to something different, I could just make that one change versus making maybe 5 or 6, however many other changes I made. Okay, before I look at the results, I have a question. So the question is, in a teaching setting, would I recommend that each student create their own CyVerse account, or is it better to use a single shared class account? So I would definitely recommend that everyone create their own account. We recommend that all the students create their own teaching account, their own account for class. And then what you would want to do once your students have created their account, you would want to get there, make sure you have their usernames, and then up in teams, you could go ahead and you could create a team, and you can say, you know, this is the so-and-so class, and then you can add members. And then that allows you to go ahead and maybe create shared folders. So everyone should have their. Then what you can do is you can choose a folder where you require everyone to put their work, and then make sure that that folder is shared. So you might want to retain ownership of that folder, but then you may want to assign your class the ability to read from that folder and write to that folder. But maybe there's even some data sets that you want read-only, because those are data sets no one should touch. So hopefully that answers your question, but you should make use of the teams and the collaborators. Also, you can make use of communities, where you can sort of link these things, because with the community, you could create, you can also curate your applications, and much of this is described in the learning site. So hopefully that answers, but go ahead and shoot me another question if that doesn't. Okay, so we have this status is marked as completed. So if I go ahead and click on the name of this, it'll automatically open up the location in the data store where that result has been displayed or output. And if I click on it, what I should expect to see is some multiply aligned sequences. And in fact, that is what I see here. So that application worked. But there is another type of application that you may want to use, one that we call an interactive application. You could have used muscle on the command line, but what is the interactive one? Let me share one slide or two, and then I'm going to come back to the discovery environment. Okay. So we have this other subset of applications, which we call interactive ones, or visual interactive computing environment. This is for using some of the nice data science applications that you might find popular. There's no restriction on data limits. It's a secure thing. Individual users can create their own environments, and you can run those for a number of days. If you need more, you can talk to us. These are great if you're working with things like Jupyter Notebooks, which I really recommend, and I love them in teaching. You could also make use of R and RStudio. So this is, again, you may want to give every student an assignment, but you can have them use their own individual R sessions. And there's no installations, so people can go ahead and just open up, click on a web link, and then they have what they need. So let me show you that in just a moment. To use one of those applications, you could search for it. So if we go back to app and I say RStudio, just as an example, you could find different versions of RStudio that may have, some of them may be pre-configured with different types of programs, but maybe you want to use something that's just sort of a basic install of R. And if I click on that one, and well, actually click on the name of it to launch it, I could also decide that there's some data sets that I want to launch with that. And then I can click launch. I'm not going to do this because I already have a number of these running. And before I get to that, there was a question about does CyVerse have the option to create interleave FASTQ files? I think you mean interleave FASTA, or interleave FASTQ files in terms of the both sets of the left and the right read. To answer that question, if I'm reading it right, okay, if you want to create an interleave FASTQ file, you need to use a software that creates interleaved FASTQ files. I can't think off the top of my head specifically of one software that does that. So if you search for the application that you would normally do and you don't find it, it's possible that we could install that. I know that some of the quality control applications interleave things. I just not use those settings, so it's probably one of the ones that I've used before, like FastP, or maybe even, I don't know if FastX Toolkit does that, but hopefully that answers your question. And if not, just share me, send another follow-up. Once I launch one of these ones like RStudio or Jupyter, what's going to happen up in the upper right-hand side is it'll tell me that it's running. And if I go ahead and click on one, sometimes it takes a few minutes for them to be available to you. So it can take as much as 5 minutes well, it can take longer than that. It really depends on if the application has a lot of data that's transferring. This one I was expecting would be done by now, but it's possible. Oh no, it's loading. So in this case here, I'm using Jupyter, which is available on the CyVerse Bice application to the discovery environment. In my case, if I go ahead and click on the notebooks, it has a whole bunch of notebooks which I've written for a classroom, in this case to do a sample RNA-seq, where they might need to do things like import data from SRA. And notebooks, which I don't have much time to talk about, are a really cool teaching platform, because especially if I'm teaching about command line, and I've verified, well, I've discussed this, we have to do more rigorous studies, but I'm sure it's true, right? We find that students, this is a nice transition to starting to learn how to Linux, because in this case, I'm using Bash. So users can go ahead and run these commands rather than having to figure out the commands all at once. And as they progress through running through these, we progressively have, more difficult things that they have to do. Here's another notebook, and they have to figure out what are the right commands and blah, blah, blah. And so they go through them. And so this is a fantastic way to teach or to use any type of data analysis. And we have different software packages that are available that way, or maybe they're working interactively with R. So you get the sense that there are a number of different features that are made available to you. And I know it can be a pain to get some of these things installed, let alone trying to have every single student install everything. And then they end up with a whole hodgepodge. And on top of that, when many of us are working remotely, you'd be very, very hard pressed to do a lot of problem solving on every individual student's individual setup. So that's there, but I'm not going to say more than that. Look at our documentation on learning that CyVerse. So here's the very last thing that I have to show you, is going to be on Atmosphere. So Atmosphere is our cloud computing setup, and you can have access to hundreds of virtual machine images. If you've not done cloud computing, I consider it an essential skill for bioinformatics. You can certainly do a lot without it, but you can do more if you know how to use it. CyVerse is one option, although there are increasingly others, both ones that you have no direct cost to, including NSF's Project Jetstream, but then also the commercial cloud. Just two pieces of vocabulary that you need to know if you've not used cloud computing before. One is this concept called an image. So let's use an analogy to explain it. Imagine that you bought a new computer, and you need to transfer things from your original computer onto your new computer. Well, there's a file that you could make. Well, you could take your files, your individual files, and back them up onto a hard drive. And those documents and files would be just documents and files sitting on a hard drive. But there's another type of backup you could make called an image. And an image file is a complete copy of everything on your system, every bit of installed software, every configuration, all of that good stuff. And the difference between a regular file and image file usually ends up being much larger, is that regular files, documents, photos, that's all they'll be on your new computer. But an image file would allow you to completely clone your old computer. You wouldn't have to reinstall that software. Even the desktop image that you had would all be there on the cloud. So it's a complete backup when you have an image file. The other thing is an instance. So in the CyVerse cloud, we physically have resources, hard drives, CPUs, RAM, and we need to provision those for you. So you ask us for a certain amount of that, and we give you that amount. And then you also need an operating system in order to make this virtual computer. And in fact, you do that, or you request that, as I'm about to show you, in about 15 minutes, you have an IP address where you have your Atmosphere instance, which is also, it is a virtual machine. There are a number of configurations that we give you. You can have up to 16 core, 128 gig machines. And some of these have quite a generous amount of disk space. You can access them with a desktop if they have one, or through the command line, and you can also share them in various ways or customize them. There are a number of reasons why you might want to do it. Many times a virtual machine is really much needed if you are doing some intensive compute and you just don't have a local computer that can do that much. There are some graphical programs that wouldn't work well on the Discovery environment. Also on an Atmosphere machine, you have full administrative or sudo access. So you don't need to ask somebody's permission to install something on a shared HPC system. So these are a number of reasons. In the demo, I'm just going to show you how you launch and connect. So I'm going to go back to the CyVerse homepage. And this time under products, well, I was going to click on Atmosphere and go ahead and say launch. When I come into Atmosphere, it's going to provide me with a list of images. Now, I happen to be logged in, so I don't have to log again. I will comment that Atmosphere access is not enabled by default. So if you signed up for CyVerse, you need to go to user.cyverse.org, and in user.cyverse, you need to look at My Services to make sure Atmosphere is there. If it's not, it'll be under Available Services, and you can click the Request Access link in order to get access to it. Okay, so from here, and assuming I'm logged in, I could search for Images like by topic, so if I typed in RNA-seq, I would undoubtedly come back with some images where there are software installed on that, right? So somebody installed a whole bunch of stuff. Looks like they have Anaconda, Callisto, Salmon, Star, a whole bunch of things that are there. Great. If I wanted to use this, I don't have to suffer the pain of installing those things anymore. I could go ahead and click Launch. And then I could decide what type of system I need in order to do this. And once I've decided that, I can go ahead and put it, in my case, in a project and say launch the instance. And in a moment or so, it'll tell me that that instance is launching. And they take a few minutes, somewhere between 10 or 15 minutes. It depends on how much data there is on that image in order for it to be spun up and usable. This will go ahead and launch. I'm not going to wait for it. Well, I want to open up a new tab, and let's just do that here because I already have stuff running. Okay, if I go to my projects and I put this in my demo project, I can see that I have something that's already running. When I have an IP address and that status is green, it means I'm ready to connect. So I'm going to go ahead and click on it. And there are a number of ways that I can connect to it. One is if I just want to use it as a command line, I can go ahead and click on Open Web Shell. And again, without installation, I connected, and I can go ahead and start typing commands and working on it. If I was actually typing correctly, it'd be even better. That's one way. This one, I believe, has a desktop, so I can go ahead and click on desktop. And I can go ahead and use this. I can see that there are folders. I could do anything that I would normally do on a computer with a desktop. It's something, this didn't exist a few minutes ago before I launched it, but now it's ready for me to use. Other ways I could connect to it, I could copy that IP address. And in my case, if I want to use my shell, I could do an SSH, my CyVerse username at that IP address. Yes, I want to connect. See if I remember my password and type it correctly. And if so, it's going to allow me to connect to it. I'm just waiting for it to decide. Hopefully I typed that correctly. Internet must be slow today. All right, so it will either connect to it or it won't. There we go. And it's the machine that I just connected to a few moments ago in Atmosphere that I could go ahead and do whatever I wanted to do. Now, I will comment that when you are connected to your Atmosphere machine, it's basically blank except for the software it said that it would have installed. If you have data sets that are on your CyVerse data store, then you'd follow the directions that are on learning.cyverse.org to go ahead and transfer those data sets into your Atmosphere image. When you're done, you can go ahead with your analysis. If you're going to come back to it, you might want to click on Shelf to put it away, and then your resources will be managed. If you look back on my desktop, I have a monthly allocation of how many CPUs and how much time I can use any of these instances. But if I need more, I can click on the Need More link to get more. It'll be refreshed on a monthly basis, but I have to at least keep an eye on that. When I'm really done, I can go back to my project and I can go ahead and delete that instance. Be careful, because once you delete an instance, you cannot get it back, and we can't rescue any data from it. So you have to be careful with that. Let's say that I've taken this instance and I've installed a bunch of software and I have really configured it in a customized way. I could also click on Image, and then I could save it such that myself, or if I make it public, my students, whomever else, would be able to also launch that same pre-configured image with all the software, all the versions, just as I originally did them, which is super powerful for creating a reproducible software environment. Okay, so we're at the top of the hour. I'm going to finish up with some slides and then encourage you, if you have any questions, to get in contact. We have just a few more moments before we're done here. The take-home message is to go back and take a look at learning.cyverse.org because we have a whole bunch of other documentation and details. We'd love for you to get in contact with us that way if you would like additional training or have additional questions. And I also want to give an acknowledgement to all of the faculty and staff at these various centers, including where I am at Cold Spring Harbor Laboratory. We are all really excited about really the thousands of CyVerse users that join us every quarter, that sign up for a new account, and that make use of the platform to do some really exciting and creative things.